All right, welcome back. We are going to continue on our journey on Chapter 5, this time moving into DNA, or nucleic acids, DNA, RNA, all of the above. So, concept 5.5, nucleic acids store, transmit, and help express heredity information. Okay, we should all kind of get the concept at this point that somehow this nucleic acid contains the information that we need. This can be linked directly back to the proteins that we just finished talking about. So the amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. Genes consist of DNA, a nucleic acid made of monomers called nucleotides. So DNA itself is a polymer. Its monomers are called nucleotides. There are two types of nucleic acids. We have deoxyribose nucleic acid, or DNA, and then we have ribonucleic acid, or RNA. DNA is more stable. RNA is less stable. DNA provides its own directions for its own replication. RNA is made from DNA. And it's through RNA that genes get expressed, or the instructions for a protein amino acid sequence come from. This process of DNA directing the synthesis of messenger RNA and then messenger RNA controlling protein synthesis, this is called gene expression. And this is kind of the central dogma of biology. So here we have double-stranded DNA in the nucleus of a cell. We are going to make messenger RNA that is complementary to one of these strands and looks like a copy of the other strand. That mRNA is going to leave the nucleus, go into the cytoplasm. It is then going to encounter something called a ribosome. A ribosome is a combination of RNA and proteins, and it is essentially a very large molecular machine. Its job is to make proteins. So what it does is reads the messenger RNA and puts the appropriate amino acid in each spot according to the directions of the messenger RNA. How this occurs is a multi-step process. We will approach this just a little bit much later on. Now, if each gene along the DNA directs a messenger RNA, and that messenger RNA is interacting with the machinery that produces the polypeptide, that protein or polypeptide is what gives us our characteristics. So an idea of gene information is DNA begets RNA, begets protein. You can even add in another areo and have trait listed out here. The connection between proteins and traits can sometimes be a little bit more confusing to understand, but it gets there in the end. Now, the components of nucleic acids is first, remember it is a polymer, and you could call this polymer polynucleotides. So it's a bunch of individual nucleotides attached together. So each one of those monomers is called a nucleotide. A nucleotide looks like this. It has a phosphate group, it has a sugar, and it has a nitrogenous base. If you talk about just the sugar and the nitrogenous base, we call that a nucleoside as opposed to a nucleotide. Tide implies that there is a phosphate group there. So nucleoside is nitrogenous base plus sugar, boom, boom, and the nucleotide, you add on a phosphate group. So The next thing we need to look at is, well, what are these nitrogenous bases? There are two words in here that you should recognize. Nitro, or nitrogen, is actually in that word there. You should recognize that there is nitrogen somewhere in these structures. Base is just as it claims. It is a base in terms of acid and bases, so it has the ability of absorbing protons. There are two main groups or two families of nitrogenous bases. One is the pyrimidines, so this is cytosine, thymine, and uracil. Thymine is only in DNA, uracil is only in RNA. Why? Well, this is one way that the cell uses to figure out what is DNA and what is RNA. It utilizes this system. You can see that these structures are related. They have a single 6-member ring. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And you can see that these all contain nitrogen within that ring. The other group is called purines, and we have two of them. We have adenine and guanine. This is a combination of a 6-member ring fused with a 5-member ring. They look very similar, but you can also see differences, such as up here we have an NH2 where we have a carbonyl group up here. Down here we have nothing. Over here we have an amino group. The other part of this is the sugars, and the sugars are different between DNA and RNA, and that is actually what contributes to the name difference, the D and the R. So this right here is something called ribose. It's a 5-member sugar, so 1, 2, 3, 4, 5. Has a carbonyl carbon and has OH groups attached to each carbon, just like a carbohydrate should. The difference over here in deoxyribose is, notice the carbon at position 2, it is missing its OH group. So it's deoxyribose. Okay, so it's missing something. This actually makes DNA more stable and RNA less stable because this OH group can go through a reaction with a longer change, which can lead to the RNA breaking apart. Oh, I just realized that right here it says DNA. It should not say that. It should say RNA. So make sure you recognize that that is RNA. I will try and fix that before I post the slides. All right, now again, these are polymers, so it's a bunch of these nucleotides linked together. This is linked together through the phosphate and sugar, creating a phosphate-sugar backbone. So here we have a phosphate, we have the sugar, another phosphate, the sugar, another phosphate, another sugar, phosphate, sugar, and that could go on and on and on. The bases stick out here to the side and are free to interact with bases of another strand of DNA or another strand of RNA, if that were the case. Notice here that if we look at the structure of the sugar, we have carbon 1, 2, 3, 4, and 5. We have a linkage between the phosphate and carbon 5, and then the next one is attached via a phosphate to carbon 3. So here we have phosphate, carbon 5, carbon 3, another phosphate, carbon 5, carbon 3, another phosphate, 5, 3, phosphate, 5, 3, phosphate. The fact that you have this flow means that DNA has a direction. Okay? So we oftentimes talk about DNA working in a 5' to 3' end. This means that this is the beginning and this is the end of the molecule. The reason we chose that as opposed to reverse has to do with how genes are expressed, and we'll get to that later on. Now, we also know that DNA is double-stranded, and it forms this thing called a double helix. So here we can see two strands that are wrapped around each other, and those two strands interact. What you might notice is one strand is in the 5', the 3' direction, and the other strand is in the opposite orientation, so 3' to 5'. So these two strands are what we call anti-parallel. They are operating in reverse directions. One DNA molecule tends to be fairly large and contain many thousands of genes. Now, DNA carries its own information on how to replicate itself. So here we have one strand of DNA, and we have various nucleotides along the way. So we have G, C, A, C. It just so happens that G's can base pair with C's or form hydrogen bonds. This is actually 3 hydrogen bonds that can form between the nitrogenous base of G and that of C. And I'll show you what those look like here in a moment. So C's would consequently base pair with G. A's can base pair with T's. This involves 2 hydrogen bonds. C with G, and then A with T again. So, as we look at this DNA, you can see that the other strand isn't the same, but it is complementary. And so if you were to remove one of the strands, you'd be able to replicate it and bring it back. Because if we were to remove the strand right here, so we didn't have this T, C, G, T, and so forth, and we only had the strand, we would know that across from this A would have to be a T. Across from this G would have to be a C. Across from the C would have to be a G, and so forth. So the fact that they are complementary strands means that all of the information for replicating itself is present in both strands. So if we split these two strands apart, we can add back in the other strand new based upon the old information. Here's a little closer look, much higher resolution, of how that interaction works. So again, here we have 5' to 3'. So phosphate, carbon 5, carbon 3, phosphate, carbon 5, carbon 3, phosphate. And down here we have 5' to 3', so phosphate 5, 3, phosphate 5, 3, phosphate 5, 3, and so forth. So it's anti-parallel. Here we have an A, and it forms hydrogen bonds with this T. You can see that this amino donates one of its hydrogens, and that links to this carbonyl oxygen. Over here, this hydrogen from this nitrogen can hydrogen bond with the lone electron pair left on this nitrogen. So A's and T's have 2 hydrogen bonds, C's and G's have 3. So consequently, C's and G's are slightly stronger than A's and T's. Here you can see another feature in here, in that there is a large gap here, but a smaller gap here. So this is what we call the major groove, and this is a minor groove. And there's a significance there. In the major groove, things could actually come in and touch the base pairs themselves. There's enough room to reach in. With the minor groove, the sugar phosphate backbone blocks access. So you can't reach in from here, but you can reach in from here and actually touch the nitrogenous bases. We will pick this up in part 2.